So one of our biggest labs that we're going to have in terms of the work that we get done is going to be lab number 7, which is where we're going to purify our MGH protein. So remember here that our MGH protein is our malate dehydrogenase that has its green backpack on that we're going to purify using the 6 histidines that are in a row. So what we're going to do in this lab is something that could take 3 individual weeks if we did it separately. But what we're going to do is split the work up into 3 teams. So we're going to have an FPLC purification team. That is a team that's going to go ahead and actually finish up the purification using our FPLC instrument, which we demoed in our previous week's labs. We're going to have another team that's going to be our spectroscopy team, and they're going to take all of these fractions and they're going to analyze them for both total protein using the Bradford assay and then the green fluorescent protein using a GFP assay. And then lastly, we're going to have an SDS-PAGE team. So SDS-PAGE is the electrophoresis scheme that we use for analyzing proteins. So fraction pooling and concentrating is something that we sort of talked about in our protein simulation lab, but it's actually something that I will do sort of on the back end here. So again, to bookend what you guys will do in lab, I will actually be doing the overexpression prior to you coming in for lab 7, and then I will be doing the back end kind of fraction pooling and concentrating so that the following week we can actually use our purified protein for a kinetic analysis. So again, just a brief overview of what's going to be happening behind the scenes by myself leading up to this lab is we're going to have a plate that we're going to streak our purified MGH plasmid containing BL21DE3 cells. That was a lot of information right there, so let me just say that again. We're going to have a bacterial plate that's going to have our BL21DE3 E. coli. That's the E. coli that's capable of overexpressing protein. And we're going to be streaking them on a plate so I can take a well-isolated colony, and then I'm going to grow that up over a series of a couple of days to get a large sample of protein or of cells here. So just to kind of highlight how we do that is I'm going to take one of these bacterial colonies and I'm going to put it in a small test tube here that's just going to have a little bit of broth. And the idea is that we don't want to take a little bit of cells and immediately throw it into a giant pool worth of media. We want to start and do what's called colony expansion. So we put it in a small kind of test tube here, maybe 3 to 5 milliliters of our liquid media. And then we're going to grow this so that we're monitoring the optical density at 600 nanometers. So what we're looking at with the optical density at 600 nanometers is actually light scattering. So the higher the density of cells, the greater the optical density at 600 nanometers. So we'll let this go for about 12 hours at 37 degrees, and then we'll have a cloudy solution as those bacteria have grown. What we'll do is then expand this by taking this 3 to 5 milliliters and we're going to put it in a 25 milliliter solution of our liquid media. We'll let that grow again, we'll expand that colony, and then we'll put that in 1,000 milliliters or a liter of liquid media. This time we're only going to let it grow not to full capacity. We're going to be monitoring the optical density a little bit more closely here because now we're getting ready to do our protein overexpression. So once we have an optical density of about 0.3, we're actually going to cool this solution to room temperature. And this is going to be unique to this overexpression of this protein. MGH does not like to be overexpressed in E. coli when they're grown at 37 degrees. And part of the reason is that the bacteria realize that we're sort of trying to slip one in under the radar and have them do some of our work. And they don't necessarily like that happens, so what they do is they trash compact that protein, they put it in what's called inclusion bodies, and it's very difficult to overexpress the MGH protein if you do it at 37 degrees. So before we actually do any work with these cells in terms of overexpressing, it's fine to get them to grow by themselves at 37. But when we're ready to do the induction, we're going to cool it to at least room temperature, and we're probably going to cool it even further, maybe down to 16 or 20 degrees rather than the 25 degrees that might be room temperature. I'll be certain to let you know what those details are. So again, over this 2-hour period while it's cooling here, The cells are going to continue to grow, so our optical density will increase. And then we're going to save a sample of these cells, and we're going to call them uninduced. So at this point, we haven't given the chemical signal for these bacteria to produce the protein that's contained within the additional plasmid that's inside their cells. So again, I'm going to say that again because that was a lot of information right then. At this point, we have a whole bunch of bacteria that are growing well. They have the MGH plasmid inside of them. And when we give them the chemical signal, they're going to start overexpressing that protein. In addition to making all the proteins that they need in their own cells, they're going to make our MGH protein. But again, that's why we needed to decrease the temperature to room temperature, because otherwise, the bacteria don't like making that protein. So again, this is what we call an uninduced cell culture. What happens then is this process of overexpression. We're going to add the chemical signal, which is IPTG. So this is a chemical that I want you to know, not the structure of necessarily, but this is a chemical that is used to initiate or induce the overexpression of proteins in our case, E. coli bacteria. So this is actually a galactose derivative. Remember, we just learned about carbohydrates. So this is the C4 epimer of glucose. So we've got galactose, and this is an isopropyl thiol derivative. So we've replaced this OH group on glucose with this isopropyl thiol group. And this is just a chemical signal that tells the bacteria, hey, it's time to take that foreign plasmid that's inside your cell and transcribe that into messenger RNA and translate that into protein. So we're going to let this go for somewhere between 16 to 20 hours. Again, I'll give you the details for what happened there after I finish this procedure. But at this point, we're going to have a cell culture that we say is induced. And several times throughout this presentation, I'm going to show you actual slides from a previous year's purification so you can get an idea of what's going on here. So again, these cells have been induced. And it's a little bit hard to see here because remember, our liquid media broth sort of has a straw color to it. But if you'll see in a couple minutes here, when we actually precipitate these cells, you'll see that they are neon green in color. And that's because they are filled with our MGH protein. Just a reminder again, the M stands for malate dehydrogenase. That malate dehydrogenase protein is tethered via a short linker to green fluorescent protein. We've called this green fluorescent protein our green backpack that our malate dehydrogenase is wearing. And then at the end of it, we're going to have a histidine tag. And again, this is going to be important because that's what we use to purify our protein in this lab. Now remember, there's lots of other things that we're viewing as contaminants. There's carbohydrates, lipids, nucleic acids. All of these are less problematic as the fact that there's tons of other proteins. These are living bacteria, so they're going to be making their own proteins to do their own cellular biochemistry. And so how do we separate and isolate what we want, which is our MGH protein, from the rest of the proteins that are there? So that's going to be the intent of this FPLC purification and the job of that research team. And then the other two research teams are going to characterize our isolated protein, excuse me, and see if we have isolated a pure sample. So once we have this induced kind of culture of cells, we're going to centrifuge it. We're going to put large volumes in these centrifuge bottles. And again, the supernatant is that straw-colored solution, really doesn't have anything that we're interested in, and the pellet has everything. These are our pelleted cells, and you can see here they are neon green, and that's because these cells are absolutely chock full of that MGH protein. What's really nice is if you're undergoing sort of a large-scale overexpression, you can actually freeze and store these cells at this point and kind of pick up from this point at another point. So it's really nice if you have a really good expression that has worked, you can kind of save and aliquot this and start from this point next time you want to make some new protein. So again, this represents a point where we can stop and continue. So again, how do we isolate and separate what we want? We've got these bacterial cells, and so here I've got a little bacterial cell here, and you can see on the inside we've got our MGH protein. So we've got essentially what looks like green snot that's right here. This is the bacterial cell, and inside the bacterial cell is what we want. So we have to lyse open these cells. So we're going to use a mechanical sonicator here. So this is what we call a dismembranator. So this sounds like, if you hate the sound when you're at the dentist's office and they've kind of got that high pitch kind of drill, this kind of sounds like that, but it sends sound waves, mechanical vibrations that are actually going to disrupt this cell wall and break open the cells and allow us to have the contents spill out. And again, our MGH protein is going to be soluble, so it's going to be in the supernatant. So again, we have this green gold, if you will, gold being like this is exactly what we want. So we've got this high concentration of our protein, and again, but it's mixed with a whole bunch of other proteins. We just happen to know that ours is there because we can see it. This cellular pellet that's down here, this is all the cellular debris. This is essentially garbage, not anything that we're interested in. But one of the things that's really beneficial from doing something where you have a fusion protein of a green fluorescent protein in your protein is you can sort of follow this process along. You can sort of see, if you were to tally up the time that we've done so far, this is almost a half a week's worth of work at this point. And if you get to this point and you don't see that you've got this neon green supernatant, well, this might be something that you're going to need to reconsider whether or not you want to keep on with the purification, because maybe you don't have your protein. So this is kind of a nice thing with this model system that we're working with that generally is not the case. You're generally not going to have a colored protein that you're going to see. You might need to do some other kind of assay, or you just cross your fingers and hope that your protein is there. All right, so again, the theme of what we're going to do here is talk about how do I isolate and separate what I want from what I don't want. And throughout this week's lab, I'm going to talk about two different methods. We're going to talk about analytical methods, and we're going to talk about preparative methods. So analytical methods are basically methods that we use to visualize things. We're going to use small amounts of sample, and we're not going to recover those samples. So when we analyze something by gel electrophoresis, again, that amount of protein is going to be gone because we're using it up in that gel experiment. The Bradford assay and GFP assays, those spectroscopy assays, we're going to take a small amount of our sample and we're going to sacrifice it to the spectrophotometer to be able to see what's going on, but we're not going to recover those samples. This is different than from the first team that actually does the FPLC. We're going to use all of our samples, and we're going to use this method for isolation purposes. So again, we're going to put all of our cleared lysate in the FPLC instrument, and then we're going to isolate the samples that we want. Again, the last piece here that we'll talk about just very generally because it's not something that you guys will be doing. This is a device, it's called Centricon, which kind of does the purpose of desalting, getting rid of excess salts, and concentrating our protein at the very end. So as we go through this, we're going to highlight the differences between techniques that are analytical and techniques that are preparative. So that's definitely something I want you to be able to think about is, those different kinds of methods. So again, overview of the experiment. There's 3 different parts here, but really what's going to be happening is we're going to be having 3 different teams doing these different parts. So the first team, the first one kind of on the scene, is going to be the team that is actually going to be finishing the FPLC purification. Like we talked about when we did our protein FPLC demo, you guys aren't going to need to use this instrument by yourself, but I do want you to know how it works. And then the FPLC team is basically going to be finishing up the purification. So we're going to be using affinity chromatography. So then that's using a specific property of our molecule, and it's going to be the histidine tag. We're going to use this preparatively to separate our MGH protein from contaminating proteins that are produced during that overexpression. So this team won't have a lot of work downstream. They're basically going to be the first on the scenes to kind of finish up and get our fractions ready for the next two teams to analyze. We are going to do these a little bit in sequence because the spectroscopy team is going to do their analytical methods and identify fractions from the purification that are both high in total protein concentration that we get via our Bradford assay, as well as those that are enriched in our own protein using a GFP assay. So do you remember when we talked about things in our protein demo, we talked about using an immunoassay that allowed us to detect our protein, and then we used a Kumasi assay that allowed us to detect all proteins. That's essentially what these two are. A Bradford assay really uses a Kumasi blue stain to stain all proteins blue, and then the MGH protein has its inherent green fluorescence, and so that's going to be looking at the characteristic wavelength of that green fluorescent protein to determine fractions that are enriched in this protein. The third team on the scene is going to be the SDS Page team. Again, they're going to use their analytical methods to separate and examine proteins in the fractions that we suspect to be enriched in MGH. So they're going to use data from the spectroscopy team. The spectroscopy team is going to say, you know what, here are the numbers of fractions that we think would be really good to take a closer look at by SDS Page. So again, that will help determine which fractions we want to pool. And as you go through this real-time, real-life sort of system, I'd like you to think back to the steps and the things that you did in our protein purification tutorial, in that simulation. And think about the times that there literally was an easy button that was just like, show me where my protein is. And we're actually going to be doing a lot of the hard work here. That's the actual real experiments. This fourth part, again, is not anything that a team is going to participate in. This is going to be something that I will do downstream. You guys will tell me, based on all of these data, what are the fractions that you'd like to pool, and then I'll go ahead and do that pooling for you. I will remove the contaminating salts and I will concentrate them so that we're ready to use them in our downstream application, which will be our last sort of actual lab that we'll do, which is a kinetics lab. And in that point, we're actually going to be looking at the malate dehydrogenase activity. So again, the green fluorescent protein activity, we really just use that to be able to see where our protein is. We're really interested in the enzyme properties of the malate dehydrogenase. So we'll investigate that in our last lab. So I'm going to go through and just spend a few minutes here talking about each team and exactly what they're doing. So these slides are set up very similarly. We'll talk about what the technique is. We'll talk about whether it's preparative or analytical. We'll talk about what you have, what you want, and then what are the contaminants? What don't you want? So the FPLC purification team is going to begin with what we call cleared lysate. Remember, we heard this term when we did our DNA mini prep, and we lysed our cells open, and then our DNA plasmid was contained in the supernatant, that cleared lysis solution. So we call that cleared lysate. So this is the same kind of a solution, and certainly there's DNA there, but this time we're interested in isolating the protein. So again, this cleared lysate really is a mixture of lots of things, but importantly, because we're interested in protein, it's a mixture of proteins. We want to isolate our MGH protein from this mixture, and everything else is really a contaminant. So how do we do this, right? FPLC purification is our method for doing this. We're actually going to be doing affinity chromatography. In a couple minutes, I'm going to remind us of some of the purification techniques that we talked about with our tutorial. And in this way, we're going to use affinity chromatography as a way to purify our protein, because that column having that nickel NTA, that nickel has a high affinity for histidines. And so by our protein having 6 histidines in a row, that's going to be a unique molecular handle to be able to take care of separating our protein from everything else. So again, no other proteins are likely to have consecutive histidine residues, so we're going to be able to separate our protein from other proteins. Anything that might be a contaminant is anything that has a histidine tag. And I'm going to kind of give you a little preview here. if we've had any protein degradation, there could be smaller pieces of MGH that still have a histidine tag. And fortunately, there's not going to be a lot of way that we can get rid of those. So we're going to talk about the kinds of things we'll do with this FPLC purification to try to minimize protein degradation. So again, 3 steps to plan an FPLC purification. Now, while the FPLC team won't be responsible for doing them, they are going to be responsible for understanding how they work and being able to report them back in their portion of the presentation. So the first thing is, again, choosing an appropriate separation technique. We'll kind of go through those steps here in a little bit. Then you need to prepare your sample, your buffers, and the column, and we'll talk about some details for what happens with that. And then you need to load the sample and then wash the column and elute your fractions. So again, you're going to have a mixture of your proteins. You'll load on the column. You hope that your sample is going to have a high affinity for your column. And even though this says the DEAE column, remember that was the ion exchange column, we're going to be using the affinity column here. You'll have an FPLC trace that you get that kind of shows the fraction numbers, the concentration of total protein as measured by the spectrophotometer, and then the saltiness of the solution as measured by the conductivity meter. And then again, we'll have our fraction collector collect all of our fractions. So just a few comments here on choosing a separation technique, and then I'm going to remind us of some notes that we had when we did our protein tutorial, our demo purification a couple of weeks ago. So remember, there's 4 main ways that we sort of think about finding something unique about our protein that allows it to separate it from other things. Maybe our protein has a molecular weight that's different from other proteins. Maybe it has a charge. And again, the charge is based on the pI of that protein, which is again a property of that molecule, as compared to the pH of the buffer you're working in, again, the environment. Hydrophobicity, how polar or nonpolar your protein might be. And then having something unique, a unique molecular handle. This is our affinity chromatography. Again, oftentimes you might use this as sort of a secondary step, sort of a fine tuning, and you'll pick something that's a little bit more broad-based. You did this with your protein purification tutorials. You maybe did a heat treatment or a salting out of your protein, and then maybe you did sort of one of these column chromatographies, and then maybe you finished up with some affinity chromatography. And while that might be something useful to do in the future, for right now, to just kind of get this done, we're going to do 1 separation techniques. And so again, that 1 separation technique that we're going to use is affinity chromatography, taking advantage of the histidine tag. Now remember, this was a super busy slide when we created it in our sort of demo lab with our protein tutorial. So I'm not going to go back through it, but I know you all took notes on this. So again, remembering that here are those 4 different types of chromatographic methods. Here's again those same 4 types. And we talked about what property of the molecule that takes advantage of, what your bead looks like, and then how you would elute your sample. Because again, if your sample is going to be interacting with the bead because of whatever the molecular property is, you need to be able to elute it and get it off. So again, here's some properties to think about with regards to MGH. And in our last lab, we sort of talked about using some bioinformatics tools to take the sequence of our plasmid, translate it into a protein sequence, and then use a protein parameter tool to determine what the molecular weight and then pI of our protein is. And while we could use this to maybe help us with size exclusion chromatography or thinking about ion exchange, we are going to be using the fact that our protein has this built-in molecular handle in that histidine tag. That's what we're going to use to facilitate the purification of our protein. So again, some things to think about. This is our FPLC team's work. A lot of sort of upfront work that they're going to have to think about and be able to understand and explain. So purification might be problematic and unsuccessful if there's an incompatibility between something that you have in your sample and either the buffer or the resin. So here are 2 things that tend to be problematic that you need to pay mind to. if there is EDTA in your sample, EDTA is a metal chelator. So if you are trying to do a his tag purification and you've got an affinity column with nickel, that's going to be a huge problem because EDTA will sequester that nickel and make your column inactive. So we tend to avoid using EDTA in any of our salts or buffers for that reason. Similarly, if your cleared lysate actually has a high salt concentration, that's going to actually present problems, particularly with ion exchange columns. Remember, when we had an ion exchange column, to get our stuff off, right? So again, here's an ion exchange. We used a high salt concentration to elute our sample. So we don't want to start with our solution being in high salt. That's going to be a problem for even getting it to bind and interact with the column in the first place. So if you find yourself having these two situations, you can either use dialysis with your sample, where you can actually try to dialyze away the salt or the EDTA, or again, these centricons can be a system that allow you to do this. This isn't going to be a situation that's problematic for us because the buffer that we use to generate our cleared lysate is both EDTA free and low salt. So that's not going to be a problem. Again, thinking about the buffers that we need to prepare, just like we did in our protein purification tutorial, you need to consider what the pKa or the pI of your protein is and what the pH is that you want to have. So again, all buffers are going to generally have some type of a buffering system. So whatever the pH is that you want to live at, you need to match the pKa of that buffer with the pH you want to be at. For example, if you want to be at pH 8.5, you're not going to want to choose an acetate buffer because acetic acid has a pKa of around 4.76. So generally, these are going to be decisions that are already made for you. Here in red are the conditions that we're going to have. We're actually not going to use a wash step. We're going to just do a two-step system here where we're going to have a loading and equilibration buffer, which is moderate pH, pH 8.1. We're going to call this buffer A. This is actually going to be relatively similar to the buffer that we used in purification of our demo proteins. Remember, we had that mixture of proteins. We had carbonic anhydrase and cytochrome C, and we had this essentially the same buffer here. The buffer that we're going to use for elution is going to be a little bit different. And now that we're not going to have a high salt buffer, it's not going to be 500 millimolar sodium chloride, we're going to be using imidazole. And remember, the purpose of imidazole, I'm going to flip back here for a second, imidazole looks like free histidine. So if our stuff is bound to the column and to this nickel because of the histidine tags that are there, if we throw in what looks like free histidine, it's going to compete for binding to our resin. And when our protein lets go, and this free histidine, this imidazole, kind of binds on, our stuff will flow through the column. So some additional things that we're going to have in, and again, these are things that are going to be important to help sort of decrease the chance that we're going to have protein degradation, particularly if we have degradation of our malate dehydrogenase green fluorescent protein. Every little piece is probably still going to have that histidine tag, which means it's going to get in the way of having a pure sample. We're going to have all these little fragments that are in there. So here's 3 other things that we tend to put in to sort of minimize and mitigate that problem. We put in reducing agents. And again, we're going to use something called DTT. It's going to be sort of a little bit of a stinky sulfur compound. And this helps maintain proteins in their native state. So see if this makes sense, right? DTT helps maintain any cysteine residues in the free thiol group. So that means we're not going to be forming disulfide bonds within our proteins. And that's important because then we're not going to be cross-linking one protein to another and kind of creating these aggregates. So this would not be something you wanted to do if disulfide bonds was important for your protein. For example, if you were trying to purify insulin, for example, not proinsulin, but insulin, you'd actually destroy this because you'd separate those 2 important pieces. So generally, this is an important thing to use, but you do need to think about what your protein is and whether or not disulfide bonds are important. We're also going to be adding in protease inhibitors. So again, proteases are enzymes that are going to break down proteins. So remember, we got this protein, our MGH protein, out of a live organism. So E. coli makes its own proteases, and we want to make sure that they don't degrade our protein. So we're going to add something called PMSF. This is phenyl methyl sulfonyl fluoride. That's what it stands for. Again, you're not going to need to remember, the structures of these, but I do want you to remember their names and what they are used for. I should have said this is the structure for dithiothietol, so that's what DTT is. So again, we've got this 4-carbon alcohol, which is essentially replacing 2 of the alcohol groups with thiol groups. Another protease inhibitor that we can add, but we talked about why we're not going to add it. Here's the structure of EDTA. So EDTA helps inhibit protease activity because most proteases require metal ions for activity. So if we sequester those metal ions, we can inhibit proteases. We absolutely don't want to do that here because the kind of purification scheme that we're doing would be absolutely destroyed by having EDTA. So we are going to add DTT, and this compound's pretty stable. PMSF actually is something that degrades fairly quickly once you put it in water. So we're going to add this just prior to purification so that it maintains its highest potency. All right, so preparing a column. You probably prepared columns in organic chemistry. So here's some important things to think about with your column. You want it to be free of cracks or any air bubbles because then your mobile phase is not going to be interacting with your stationary phase at that point. We want to pour it by preparing a 50-50 mix of resin and whatever our running buffer is. So again, our columns are going to be about 5 milliliter columns. So we'll have 5 milliliters of our resin and then 5 milliliters of running buffer to make this 10 milliliters of slurry. You pour it in and you let it settle by gravity. You can try to accelerate this process by kind of pulling it through or trying to accelerate it, but generally the best columns are going to be one that we allow to settle by gravity. Unless it's a virgin resin, meaning it's never been used before, we want to wash it prior to use by both a high salt buffer and then equilibrate it with our running buffer. So again, we're going to use a high salt buffer because if we've used this resin before, and this is an important note, that resins can be reused. If we're going to use this resin again, we want to make sure that we've knocked all contaminating proteins off, particularly if we've used this for another his-tagged protein. We don't want to have those mixed together because we won't be able to separate those. So we run high salt through to kind of wash the column, but then we need to make sure to get rid of all that salt so that the new stuff can bind. So we need to equilibrate it with that low salt buffer. I'm not going to spend too much time talking about this again because we spent some time in our FPLC demo in our previous lab sort of talking about what this sample loop does. So I've kind of got shown here in either green or red what the pathways are. Remember, this is sort of like a 6-way system that, depending on how this knob is turned, determines whether we have our standard buffer route. And in this case, this is where our mobile phase is not stopping at the bus stop to pick up our passengers, okay? Or we've got our loading position. Now our pump is going to stop at the bus stop. And again, we don't kind of show that long tube here, but then stops at the bus stop and picks up those passengers to take them through our system. So this is, again, how we're going to load our sample. So we went through that in some good detail when we did our protein demo. So we're going to have the same thing. And again, this will be something that'll be done well before the FPLC team comes on the scene. They're going to come in probably for the last 15-20 minutes of the purification, just so that the other teams then are ready to start using those fractions for their analyses. So let's see here. I think I have, not yet, but we're going to have a couple of videos that we'll have here to just kind of show what's going on. So here I'm loading the sample. So again, I've got our sample that we're putting in the syringe, and we're going to load it into our sample loop here. So remember when we had our demo purification, we had a fairly short sample loop because we only had 1 milliliter of our sample protein. In this situation, we're going to have a lot of our a lot of our MGH protein, so we have a much larger sample loop. And let's see, I think these might be, oh, not videos yet. So we are going to then have, let me just make sure. Yeah, these are not videos yet. So we are going to have, and this is actually a DEAE column, so chemistry and the FPLC operation is going to be the same. It just happens to be an affinity column or an ion exchange column rather than an affinity column. So what's going to happen is our sample is going to kind of come into the column here. It's going to kind of flow into this headspace here, and it's going to start flowing through and interacting with our column. Here we have our videos here. So here we're just going to show a little video here that will highlight sort of what happens here as our sample continues to migrate through the column, and we're going to have our stuff kind of bind to and stick with the column. So you can see high concentration of protein here. You know when you have two solutions that you mix that are very different in their concentration, you see these little currents that we have here. And that's because we have two solutions that have very different concentrations. So we had our running buffer, and now we've got our protein that's here that's being loaded on the column. So again, after I've finished loading, I'm going to turn my my sample loop here to disengage my sample loop. I don't want my bus continuing to stop at the bus stop once we've already picked up and loaded all of our passengers. So a few more videos here. This is just showing the FPLC, and it's showing the trace that we're going to be acquiring. It's already started collecting the first fraction, and what we're going to see here is we're going to see this blue line, which represents the absorbance of protein, is going to increase dramatically as we start having our contaminating proteins that did not adhere to our column make their way through the column and to the spectrophotometer. So again, our blue is increasing as we are having higher and higher concentrations of protein that are coming off the column because they're not interacting with the column. And remember, I talked about this in our demo. I usually like to have a tiny little bubble that I have from the last part of my sample. And this is, essentially the last passenger that was picked up. And now I've got kind of pure mobile phase that's kind of coming back here. So this allows me to kind of know exactly when my sample ends and just the mobile phase behind it begins. So again, this is going through and going through to the column. And once I know that all of my passengers have been picked up from our bus stop here, we'll make sure to switch that so that we don't have to be running through anymore. So again, now I'm switching that so that I make sure that I'm not having my mobile phase stop at the bus stop anymore. All right, so here's going to be another video just showing, we're going to see a little air bubble that's going to come through here, and this is going to represent that little air bubble that was the last of it was right at the back end of my last passengers. And then we'll be able to see a line of sort of green move down here as the solvent head now becomes our mobile phase rather than the sample that we're loading. This video is a little bit longer because I go back and forth between the instrument here and the column. So there you can see the bubble coming through. So that's the last little bit of our sample that's been loaded. Now it's just going to be pure mobile phase that's coming through. So we'll start to see, you can see here a line. There's a line of where our the last bit of our sample is, and it continues to sort of make its way down through the column. So at this point, we continue to have sort of some contaminating proteins that are not interacting with the column come through. And so we're going to continue to see the absorbance, that blue line on the FPLC trace, increase. So again, the last little bit, this is kind of the line between our solvent or our mobile phase and then our sample that we were loading. And what's kind of neat here is you can see that the green is sort of consolidating here. We're seeing that all of our protein is sticking to our column, which is what we want. So here is the little video where we're going to show the concentration in protein as measured by UV is going to be increasing significantly as those contaminants continue to come off the column as we've finished loading our sample. We're not very far into our run here, only about 10 minutes in, and we've sort of completely loaded our sample. So again, focus here, we can see that this is pure mobile phase, it's up here. Our column is nice and green. Actually here over here you can see the nickel column for affinity. It's actually bluish in color. You can see here's the huge sort of spike, significant, we talked about 2 being sort of the maximum in terms of absorbance units. So these are all contaminating proteins that have come off. So one last little video here is going to show now this is actually going through the elution phase. So we're increasing the salt concentration here. And for this demo, I actually had the gradient increase fairly sharply so that we have a very steep increase in the concentration of, in this case, because this is ion exchange, I did have salt that we were increasing the concentration of. And that's just so that in this video, we can quickly see all of the protein coming off at one time.
I'm literally watching it let go of the column. Just a second ago, all the green was at the top. And as the salt gradient is going through here, our green fluorescent protein is going to start eluting off. You can even see as we're going here, it's moving further down our column. More white on top. And I think very shortly we'll see some green start to come through our column here. I'm watching the absorbance peak greatly here, so it must be coming off. You can definitely see that it is eluting off the column right now. There it comes. Definitely can see that protein coming off. It's almost like our tubing here is neon green. You can see that it's coming through here. Probably shortly we're going to see our salt gradient bump up as well. Just like that, our green has come off the column.
So now getting to some of your pre-lab questions. So this is just the answer here for the first pre-lab question, which asks you to draw what you'd predict for a plot of conductivity as a function of time given the following sort of buffer conditions. And again, telling you what the buffer concentrations are. And again, question asking you to draw a predicted FPLC trace showing results from the conductivity meter. So feel free to pause the video at this point, kind of make sure that you can see these different points. I'm just going to kind of highlight them briefly here. When we have buffer A, low salt, low conductivity. For the next 20 minutes, we go from a 0 to 70% gradient of our high salt buffer, buffer B, and so we have a 20-minute sort of step up here. And then over the next 2 minutes, we quickly ramp from 70 to 100% B. Then we stay at 100% B for 2 minutes, and then we go back to 100% A for the next 5 minutes. So again, hopefully that makes sense. We talked about something like this already in lab number 6. So again, here are some details for the program if they've changed, or if they do change for this, I'll make sure to let you know. But in general, the program that we run is that we're going to collect all the fractions. We're going to spend 3 minutes at each fraction. The flow rate's going to be about 1 milliliter per minute. We're going to spend 30 minutes at buffer A, and then between 30 and 2 hours, we're going to gently go from 0 to 100% of buffer B. And so what we can see here is a lot of contaminants come off, and then we can see kind of a slow eluting of some different protein components here. And again, here you can see we've got an increase in our salt gradient. So yours is going to look a little bit different because this was done for an ion exchange chromatography. So again, fractions are going to be collected every 3 minutes, flow rate of 1 milliliter per minute. So each tube has 3 milliliters of solution. So what have we accomplished when we're finished with our FPLC team? Well, we've separated or resolved our proteins. That's desirable, right? The extent of separation is going to be inversely proportional to the steepness of our gradient. You know, if I blow everything off the column in 2 fractions, I really haven't done a good job separating things, right? But a challenge is that our proteins become more diluted, right? If we started out with a 5 milliliter protein sample that we loaded on our column, and we have what looks like to be about 40 different fractions here, each being 3 milliliters, we've got 120 milliliters of solution. So in essence, we've just diluted all of those proteins out over a much larger volume. So again, that extent of dilution is also inversely proportional to the steepness of the gradient. If we've got a shallow gradient, it's ideal for maximum resolution, but then we're going to have a large volume to accomplish this. We're going to have lots of diluted fractions. So it's sort of kind of trying to find that sweet spot between having enough resolution that you separate what you want from what you don't want, but not diluting things so much that you really have a hard time working with it in your downstream applications. All right, so I know this video is getting to be pretty long, but again, this is why we're sort of flipping it so you can potentially watch it in maybe some chunks. So that was the first team. That was our FPLC team. We're now moving on to our kind of spectroscopy team. So the next 2 teams are going to be involved in analyzing the fractions that the FPLC team collected. So again, this is going to be an analytical method. This is going to be something that we're going to utilize to kind of visualize what's going on. We're not going to take the entirety of every single fraction and use it in the spectrophotometer, because then we'd be wasting it. We really wouldn't have any sample left then to work with. So we've got lots of fractions. We want to identify those that are enriched in our protein, and we want to know what ones have a lot of contaminants because we'd like to eliminate those. And so again, proteins that are contaminants are really anything that doesn't have a histidine tag. Because we use that affinity chromatography, we should be able to have only those proteins that had eluted in the column or eluted off of the column later on should be ones that have a histidine tag. I apologize here that some of the FPLC demo videos that we showed were actually for an ion exchange. The principles are all the same, but that was why we eluted with salts. We're going to be eluting for the FPLC team with imidazole. So just make sure, I wanted to highlight that difference. So again, spectroscopy is how we're going to be doing it. You guys actually made a figure like this when we did our data analysis lab way at the beginning of the semester. You may not have known that this is what it was going to be used for, but when you did a double Y plot, you did a green and a blue trace. Remember, the green was sort of looking at where the green fluorescent protein is. The blue is looking where all proteins are. So again, we're going to call this work by the spectroscopy team sort of looking for blue and looking for green. So looking for blue is what the Bradford assay is going to do. So by measuring the absorbance at 595 after we add a specific reagent to fractions, we're going to be able to determine the total amount of protein in each fraction. And then we're going to use the absorbance at 492, or actually I want you to use whatever you found in our boot camp lab where you guys had to do a wavelength scan of the green fluorescent protein. So you're going to need to go back in your lab notebook and find that piece of data to know what wavelength you want to use to measure the absorbance for green fluorescent protein. It should be something around 490 nanometers, but you guys will use your specific number. And again, what we're hoping to do here is separate fractions that have contaminants from fractions that have our stuff. And remember, in our tutorial lab, we talked about sort of these mutually exclusive parameters of yield and purity. If you're going to have a high yield and essentially save everything, you're compromising purity in order to do that. So we'll have to think about, and the spectroscopy team is going to have to make a determination of what fractions do they think are going to be enriched in our protein and probably not having a lot of contaminants. So again, we call this looking for blue and looking for green. We're going to be using essentially the microplate reader. So we're going to be doing spectroscopy, but rather than using the single cuvettes that we've used like in our boot camp lab, we're essentially going to be use a 96 well plate, which is going to analyze 96 samples at once. So what you'll do is you'll actually put some of your fractions in these wells, and then you'll add a reagent that has Kumasi Brilliant Blue. This is the same dye that we talked about in our protein purification tutorial. You remember when you clicked on the gel and it would click on Kumasi Blue, would show you all proteins, and then you'd click on the immunoblot and that would just show you your protein. So again, this is sort of the equivalent of that, where we're going to be using this Kumasi Blue to show us where any protein is. And then when we look for green, we're going to be using, and again, here's just a different lab group's absorbance that they had for that year. So again, usually around 490 might be where this is. But you're going to use whatever the wavelength is for the maximum absorbance for your green fluorescent protein, and you're going to find which protein fractions are enriched in green fluorescent protein. So again, looking for all proteins, looking for my protein. So two terms that I kind of want to highlight here, and we've sort of mentioned these terms absolute and relative before. Do you remember when we talked about our statin dilemma and we talked about sort of absolute reduction in risk versus relative risk reduction? So absolute and relative are two terms that are really important to understand kind of in the scientific community. When we talk about an absolute amount of protein that are in each fraction, we're talking about an actual number. And again, we're going to use an assay that uses a standard curve to compare the amounts of a standard protein to that within our FPLC fractions. So again, when we have this blue line here, we're going to be able to use these data and we're going to be able to get an absolute number of milligrams of protein that's in each fraction. So again, we'll be able to know for each of these little dots here exactly how much protein that corresponds to. And so we can actually compare these and we can know exactly how much is different between each of these fractions. And this is different, for example, with what we do with our green fluorescent protein. In this case, we're not going to be using a standard curve. This is just going to be something that shows a change in the amount of greenness, if you will, in our solution. So it's going to show us the relative amount of MGH protein that's between different fractions. So again, we'll be able to see that these fractions have different amounts of green fluorescent protein, but we can't use this green assay to determine the amount that's there, just that they're different and that one is greater than the other. Again, to highlight, because we have a standard curve that we're using for our Bradford assay, we'll be able to use that standard curve to know, for example, that this amount of blueness corresponds to this absolute amount of protein. There's nothing that we can do with this green data point to tell us exactly how much green fluorescent protein is there. We just know that relatively it's more than we see here and here. Prelab question number 2 asks you to predict and draw what you'd think for a hypothetical double Y plot. And again, consistent with the method of purification that we're going to be using, which is again going to be affinity chromatography. So again, we predict that there are going to be, and shown in blue here is total protein, some weak binding contaminants that come off. And then we might, predict that early on, maybe some stronger binding contaminants might come off as we start increasing the salt. And then finally, we'll get to a high enough, I shouldn't say salt here, I apologize, that we get to a increasing concentration maybe of imidazole, and maybe that allows some stronger binding contaminants to come off. And then finally, we're going to have our protein come off. And then again, this situation was asking about, so make sure that yours looks a little bit different. This is drawn for thinking about ion exchange. So again, things that are going to have a similar PI will come off with MGH using ion exchange chromatography. We're going to be using affinity chromatography. So this would be MGH and then contaminants that also have a histidine tag. So again, this is important because this could be proteins that are degraded, MGH sort of degraded proteins that still have a his tag. So make sure you note that difference when you make your own pre-lab, so that pre-lab answers, so that you don't exactly copy this word for word, because this is what's going to be a little bit different, because we're not going to be doing ion exchange, we're going to be doing affinity chromatography. Just a little bit of the plate reader here. This is an instrument that we have downstairs. So again, a spectrophotometer that can analyze up to 96 samples at once. You can look at up to 6 wavelengths at one time. You can heat, shake, do a time course. It's a pretty amazing instrument. We'll be able to probably get a chance to use it a little bit more in Biochem 2 as we start doing some analyses of mutant proteins. So again, looking for blue, I just want to kind of highlight how this works. So again, the Bradford assay is a colorimetric assay that looks at the addition of this dye to a solution that contains protein. So when that solution contains protein, it turns what they call a brilliant blue. You guys are going to generate a standard curve using a protein that has a known concentration. So again, we see an increase in the blueness as we increase the concentration of protein. This is the exact same data that you guys worked with when you had your data set that you did for Bootcamp 101 when we did data analysis and you had to make a standard curve. So now you're going to be doing it again, except now this time you're generating the data. So again, third pre-lab question asks you to draw a predicted plot for your calibration curve. So again, measuring the absorbance at 595 is a function of the concentration of our standard protein. We should see a linear increase in the concentration or the absorbance as we increase the concentration. So again, we're going to obtain these different concentrations of solutions by doing sort of a serial dilution of a stock protein. So not like we had with our calculations. So don't worry, we're not doing that. There's very detailed directions for how you do that. You get the equation of this straight line, and then you're going to use that equation to determine the concentration of protein that's in any of your samples. And again, remember, this is going to be total protein. So don't just copy this right here. This is actually not correct here. Solve for unknown quantitative value of all proteins by comparing to its standard curve. So again, fraction analysis is going to be done. You're only going to take a small amount to look for blue. So 10 microliters of each of your fractions. Okay. And then again, relative amounts of each protein could be determined by peak heights, but we're going to use our standard curve to kind of figure out the absolute amount of protein. Okay, so again, this is going to be important for looking at total protein. And again, total protein, we will be able to get absolute amount milligram quantities of our protein by using our standard curve. For the green, right, because we're just looking at relative amounts, we're just going to be looking for the fractions that are most enriched in our protein, the green protein, so that we can kind of eliminate those from ones that still have protein but probably not ours. So something a little bit different here, and the spectroscopy team will get a workflow sheet that'll go through the details so you know exactly what you're going to be doing. We're going to use a much larger portion of our fraction, 200 microliters, in order to be able to get a strong enough signal to see. All right, last team, team number 3 is our spectroscopy, our SDS page team. This is, again, another analytical method. It's still a separation tool, right? We're going to separate our proteins on a gel matrix so that we can see what's there, get a better visualization of what's there. And again, it's going to help us identify fractions that are enriched in the protein that we want and identify ones that have a lot of contaminants that we probably don't want. So again, gel electrophoresis is going to be how we're going to do this. SDS page separates proteins according to their size, their electrophoretic mobility. SDS basically denatures. This is sodium dodecyl sulfate. This is a soap. So it denatures all proteins, makes them rod-like in their shape, and coats them with a negative charge. And this allows every protein to sort of migrate through our gel, and it's going to separate them based on size. So again, we want to make sure that we eliminate any fractions that have contaminants. So by doing this gel analysis, we're going to be able to have a visualization of exactly what's in each fraction. This is like when you did your protein purification tutorial. And remember, to tell whether or not you were done with your purification, you had to get to one spot by your SDS page. So this is essentially what we're looking at here. Can we analyze fractions? And we're going to take the fractions that really only have, in theory, one spot. That's probably not going to be the case, but we're going to want to make sure we take ones that are most enriched in our protein and have the least amount of contaminants. So again, we'd expect that these early fractions that come off, there's going to be lots of blue, but not a lot of green. And that is what we see here on our double Y plot. Once we elute off our fractions, right, we can already see that there's a lot of green here. There's still going to be lots of blue, because remember, that's protein that's there. But by looking where the green is, we can help say and realize that, you know what, these fractions are going to be the ones we're interested in saving. This guy, there's a lot of blue that's over here, but we're not interested in that because there's really not as much green that's there. So probably a different contaminant, something that we're not at all interested in. So again, this SDS page team is going to accomplish, a couple of things. They're going to help determine the fractions that contain protein. Again, the spectroscopy team is going to help us determine which ones have a lot of total protein and which ones are enriched in our protein. And so this SDS page team is going to help kind of refine this. So again, spectroscopic analysis will determine the fractions that are enriched in total protein and enriched in our protein. And then the SDS page team will take those specific fractions, analyze them a little bit more closely, and see what we might want to save and maybe we want to discard. So 4th pre-lab question here. And again, this pre-lab answer is based on ion exchange chromatography. Make sure that you change your answer. The pre-lab question is going to be written for affinity chromatography, but you're going to want to draw a hypothetical SDS page gel. And again, the samples that are included in the lab are a protein marker. When we have our uninduced sample, there's basically just a whole bunch of these cellular proteins that are here. Our protein isn't here. The induced sample is going to have an enrichment in the MGH protein. Again, the cleared lysate is basically going to be the soluble parts of what's in the induced cells, so this is going to look very similar. The flow-through, this is essentially what came off in our early fractions here, these guys right here. We'd predict that there's a lot of the contaminating proteins, but hopefully not our protein. Now again, one way that you could see some here is if we had way more protein than we had seats or spaces on our column. We would have overloaded our column then, and we would have had some of our protein come through just because there wasn't a spot for that protein to sit on the resin. We always want to make sure we load an authentic sample. So again, you'll have an authentic sample SDS page team to analyze. This is going to allow you to make sure that you know exactly what is the sample or the protein that we're interested in. And then again, the spectroscopy team is going to help identify the 7 to 14, or in lane 7 to 14. So there's going to be about 7 to 8 fractions that the spectroscopy team is going to ask the SDS page team to analyze. So based on the spectroscopy team's recommendation, the FPLC team is also going to weigh in on this. You guys are going to determine what fractions we want to analyze here. And again, some of these fractions that maybe are enriched in protein will confirm that, you know what, not enriched in our protein. Nope, not here either. This is a fraction that we'd really like to save. Not a lot of contaminants, but very enriched in our protein. So again, the SDS page team is going to be sort of the final fine resolution analysis of what's going on to help determine what fractions you guys will tell me you want to mix together and utilize for the kinetics experiment. So the fraction pooling is actually something I'll do. I'm going to tell you the details for how it happens, but this is something that you guys won't be responsible for doing. You just need to tell me what fractions you want to pool together. Just to highlight, this is another preparative method. This is going to be something where we're throwing all our eggs in one basket. We're pooling our samples together. We're choosing what samples we say are ones we want to move forward with. And the fractions we want to move forward with are fractions that are enriched in MGH. So again, what we want is a small volume of solution that's low in its salt concentration and then high in MGH. Remember, the only way that we got these to come off of the column is we had to have really high concentration of imidazole. So that's not something we're interested in having moving forward. So we need to get rid of that. And ideally, we'd like to concentrate our samples so we have a high concentration of protein. So we need to get rid of any salts that might be there. And we'd like to remedy the fact that our protein might be fairly dilute right now. So again, how this works, one method is you can use these centricon devices. So basically we're going to do a series of centrifugation steps and dilution steps. So centrifugation steps concentrate our protein, dilution steps dilute out the salts. So we'll kind of walk through how this happens here. But again, we want to be concerned about contaminants that were there, and maybe these salts might be contaminants to downstream applications. Right? So again, this phase is as done as it's going to be right now. Again, our protein could either be ready for the next step in purification, or as we're going to do, it's going to be kinetic analysis in our next lab. So just to highlight here what sort of happened, right? SDS page, that fine resolution work by that team, determined and confirmed fractions that had a lot of protein, that were, again, the blue fractions in the Bradford assay by the spectroscopy team, but then also refined and said, you know what, more than just having a lot of protein, it has a lot of R protein. So again, the SDS page team refined that. So your last pre-lab question really has to do with thinking about doing some dilutions. And I don't have the answer sort of written out here. I'm going to go through on the next slide sort of how this works here, and then we'll come back to this slide. And I know this looks like a really busy, scary slide, but just stay with me for a second and see if this makes sense. Imagine that we've pooled 5 fractions that each have 3 milliliters in them. Hopefully it makes sense that we'd have 15 milliliters of total solution. Now in this example, I had a salt purification. So we're going to have a high concentration of sodium chloride, but you could just as easily imagine that this is a high concentration of imidazole that we have here. And maybe the protein concentration that we have here is, say, about 5 micromolar. So this is what we have when we mix, looking for this slide. Oops, where is it? This slide. When we mix these 5 tubes together, we're going to have 15 milliliters of a solution that's high in salt, or in our case, imidazole, and has a given protein concentration. Okay? So let's see if these steps make sense. I'm going to do a centrifugation step. Okay? What's going to happen is I'm going to take this 15 milliliters that was in here. I'm going to force it through this membrane. There's a little membrane that's down here. This membrane is small enough to allow salts to go through, but it's so small that our protein can't get through. So salts will flow through, and we don't change the salt concentration. So our ion concentration stays the same. We went from 15 milliliters down to 1 milliliter. So that meant we did a 15-fold concentration of our protein. So as it says here, centrifugation steps. There's a dialysis membrane there that has what we call a molecular weight cutoff. So a molecular weight cutoff of 30,000 means only things that have a molecular weight less than 30,000 Daltons. So water, small solutes can pass through. Protein can't pass through. So now that same amount of protein that was once in 15 milliliters is now in 1 milliliter. So it's become more concentrated. If you need to, go back and replay that so you understand what's happening in the centrifugation step. No change in protein or in salt concentration. Concentration of protein. The next step is we're going to add a solution of what we wish our protein was in, the desired buffer solution. So here it's going to be essentially a 10 millimolar tris pH 8 solution, no salt, no additional ions. What happens is, just like you'd imagine, if we add a dilution to it, it's going to dilute everything. So the salt gets diluted by a factor of 15. The protein gets diluted by a factor of 15. So the protein gets back to what its original concentration was, and then our salt solution here gets diluted. So we're going to have subsequent steps of centrifugation followed by dilution. Centrifugation followed by dilution. Every time we do a centrifugation, nothing happens to the ion concentration, but we concentrate our protein. When we do a dilution step, we dilute both the ion concentration and the protein concentration. So we're going to go through a series of these so that we eventually get down to the concentration of protein that or the concentration of salt or of ion that we want. So hopefully that made sense. So we're going to be kind of pairing centrifugation, dilution, centrifugation, dilution. You're always going to be doing both of those together. The last step that you're going to want to do is just the centrifugation, not the extra dilution. So imagine each step has kind of an A and a B part, the A being centrifugation and the B being dilution. So we do first, we'd have a 1A and a 1B. Then we'd have a 2A and a 2B. Then a 3A and a 3B. And then the last thing we're going to do is a 4A. We're going to do a final centrifugation because we've already gotten the salt concentration to be what we want, but we want to go back and use that centrifugation to concentrate the protein. So notice the centrifugation step involves concentrating the protein. The dilution step dilutes everybody. All right, so let's come back here and look at what we're asking here. We'd want to know how many times we have to go through these dilutions to take a salt concentration that was 1 molar and get it down to 0.1 millimolar. So I'm going to leave this one up to you guys. Talk to a neighbor if you want. See if you can figure out how many times do we have to go through 1A, 1B, 2A, 2B, 3A, 3B, and so forth. How many of those do we need to go through before we're going to take our starting 1 molar solution and end up at 0.1 millimolar? So wow, I know this was a really lengthy pre-lab, but that's why we're flipping it outside of our lab experience and having it be something that you'll do in our flipped classroom experience so that we'll actually use in lab time, but it's going to be done on your own time.